background. Background. Okay, na? మా ఫ్యాన్ అయ్యావా ప్లీజ్. Yeah, Shreya ready. Good morning, everyone. So in previous class, continue a double fertilization. Double fertilization. In sexual reproduction of flowering plant lesson, continue topic, that is double fertilization. What is a double fertilization? See, imagine this is the embryo sac. In previous class already we discussed. This is the embryo sac. Embryo sac is also known as female gametophyte. The structure of embryo sac already we discussed in previous class. So in the structure of embryo sac, 3 cells present towards the anterior portion and 3 cells present towards the posterior end of embryo sac, and 1 large cell present in the center of embryo sac. Here 3 cells are present. In that, these 2 cells synergids, and this cell is called as X. 3 cells present towards the anterior portion of embryo sac. These 3 cells, in that 3 cells, 2 cells are called as synergids. Between 2 synergids, 1 egg cell is present. These 2 synergids and 1 egg cell collectively called as egg apparatus. Already we know. 2 synergids and 1 egg cell, both collectively called as egg apparatus, and 1 central large cell present. That cell is called as central cell. 1 large cell present in the center of the cell. The center cell is called as central cell. And 3 cells present towards the lower end or posterior end of embryo sac. These 3 cells collectively called as antipodals. We know this one. Already this structure we studied in previous class only. And listen carefully. Generally, synergids present in haploid condition, XL present in haploid condition, and central cell present in diploid, antipodals present in haploid. Polarity is the compulsory. Synergids present in haploid condition, XL present in haploid condition, central cell present in diploid condition, and 3 antipodals present in haploid condition. Why central cell present in diploid condition? Why central cell present in diploid condition? Because of the central cell consists of 2 polar nuclei. That's why it is present in diploid condition. And see, listen, this is the structure of embryo sac or female gametophyte. Now, what is our topic? Actually, what is our topic? Double fertilization. See, listen, imagine this is the pollen tube. Just imagine. This is the pollen tube. In pollen tube, how many male gametes are present? 2 male gametes are present. These are the male gametes. So, the pollen tube now bears 2 male gametes. It is very clear. See, during the process of fertilization, this pollen tube is enter into embryo sac through one of the synergid, out of 2 synergid. For example, this is one synergid and this is one synergid. Through one of the synergid, pollen tube is opened into the embryo sac. Then after, whatever 2 male gametes are present, the 2 male gametes are released into the embryo sac. Out of 2 synergid, through one of the synergid, pollen tube is opened into the embryo sac. Then after, a small pore formation takes place. A small pore formation takes place in the tip of pollen tube. Through that pore, these 2 male gametes are released into the embryo sac. Now these 2 male gametes are opened into the embryo sac. See, listen, what is the next process? See, listen. Imagine this is one male gamete fertilized with egg cell. Out of 2 male gamete, 1 male gamete fertilized with egg cell. First male gamete, I am writing here, first male gamete fertilized with egg cell. Egg cell is also act as female gamete. Egg cell is also act as female gamete. Out of 2 male gamete, 1 male gamete is fertilized with egg cell to form zygote. Results to form zygote. First male gamete fertilized with egg cell or female gamete, result it makes zygote. And second male gamete, this is the second male gamete fertilized with central cell. The second male gamete fertilized with central cell to form pen, primary endosperm nucleus. Pen full form, primary endosperm nucleus. Simply it is called as pen. Out of two male gamete, one male gamete fertilized with egg cell to form zygote. And second male gamete, the second male gamete fertilized with central cell to form primary endosperm nucleus pen. And listen, here male gamete by default haploid condition, egg cell is also haploid. Haploid and haploid, that's why zygote present in diploid. Haploid male gamete fertilized with haploid egg cell to form diploid zygote. And here also male gamete is haploid. Second male gamete is also haploid, but central cell present in diploid. Central cell present in diploid. Haploid male gamete fertilized with a diploid central cell to form triploid primary endosperm nucleus. To form primary endosperm nucleus. Okay? So haploid male gamete fertilized with haploid egg cell to form diploid zygote. Second haploid male gamete fertilized with a diploid central cell to form triploid primary endosperm nucleus pen. See, listen carefully. Listen. So formation of zygote, that process is called as syngamy. What is syngamy? Male gamete fertilized with egg cell. Male gamete fertilized with egg cell to form zygote, that process is called as syngamy. Second male gamete fertilized with central cell to form pen, that process is called as triple fusion. First male gamete fertilized with egg cell to form zygote, that process or that fertilization is called as syngamy. And second male gamete fertilized with central cell to form primary endosperm nucleus, that process is called as triple fusion. Listen, this syngamy is one fertilization and triple fusion is the one fertilization. Mean this is one fertilization and this is one fertilization. This fertilization is called as syngamy. This fertilization is called as triple fusion. So this is one fertilization and this is one fertilization. That's why these both phenomena collectively called as a double fertilization. Syngamy and triple fusion. Syngamy is one fertilization, triple fusion is one fertilization. These two fertilization collectively called as double fertilization. That's why double fertilization is equals to, I'm writing like this, double fertilization. Double fertilization is equal to syngamy plus triple fusion. Double fertilization is equals to syngamy plus triple fusion. And this double fertilization is very common in case of angiosperms. Very important. It is not in gymnosperm. It is very common. Double fertilization is very common in angiosperm. What is double fertilization? Syngamy plus triple fusion. And see, this syngamy was discovered by the scientist Strasburger. The discovery of syngamy, credit goes to Strasburger. And the discovery of triple fusion credit goes to Navashin. And the double fertilization credit goes to Navashin only. Expected question, Singami. Singami discovered by the scientist Strasburger, triple fusion discovered by the Navashin, automatically double fertilization discovery also credit goes to Navashin. See, listen, because of Singami, what is the product? Zygote. Because of triple fusion, what is the product? Pen. Because of Singami, what is the product? Zygote. It is the diploid. And because of triple fusion, what is the product? Pen, primary endosperm nucleus triploid. The product of Singami, zygote, it is the diploid. The product of triple fusion, pen, it is the triploid. In later stages, this zygote is developed into embryo. The zygote developed into what? Embryo. Pen developed into endosperm. In later stages, diploid zygote is developed into diploid embryo, and triploid primary endosperm nucleus is developed into triploid endosperm. In the latter stages, by repeated mitotic division. By repeated mitotic divisions. Repeated means many. By repeated mitotic division, diploid zygote is developed into diploid embryo, triploid pen developed into triploid endosperm. How we will see? And listen here one important point. This endosperm provide the nutrients to developing embryo. What is the function of endosperm? This endosperm provide nourishment. Endosperm provide nourishment. It provide the nutrients to developing embryo. That's why endosperm is considered as the nourishment tissue. Means nutritive tissue. Why endosperm is considered as the nourishment tissue? It provides the nourishment. Means it provides the nutrients to developing embryo. That's why only endosperm is considered as the nourishment tissue or nutritive tissue. So this is the information which related to double fertilization. Very important topic in sexual reproduction in flowering plant. So next stage is what happened? Zygote developed into embryo, pen developed into endosperm. How zygote developed into embryo, now we will discuss in the topic of embryogenesis. Next topic. Embryogenesis. Embryogenesis. After double fertilization, embryogenesis. Okay. See, listen, in case of angiosperm, after double fertilization, in case of angiosperm, after double fertilization, ovary developed into fruit and ovules are developed into seeds. In case of angiosperm, after the fertilization, means after the double fertilization, ovary developed into fruit and ovules are developed into seeds. The seed topic also we have. So now before going to seed, first we are going to discuss with embryogenesis. Genesis means birth or formation. What is the meaning? Microsporogenesis, megasporogenesis, embryogenesis. Wherever genesis starts, genesis means birth or formation. And birth or formation of what? Embryo. Birth or formation of embryo. Birth or formation of embryo. That process is called as embryogenesis. From where embryo was developed? Already just now I discussed. See, because of syngamy, which one is formed? Zygote. It is the diploid. This zygote undergoes a repeated mitotic division. The zygote undergoes repeated mitotic division. Finally, it is developed into diploid embryo. Here, mitosis is occurred. That's why diploid and diploid. If meiosis is occurred, chromosome number is reduced to half. But here, what happened? Mitosis. That's why chromosome number in the parent and chromosome number in the offspring is the same. From where embryo is developed? Embryo is developed from where? Zygote. By repeated mitotic division, by many mitotic division. The zygote undergoes many mitotic or repeated mitotic division. Finally, it is developed into embryo. But here, zygote is not developed into embryo directly. Zygote not developed into embryo directly. Some stages are there. For example, listen. This is the, for example, this is the zygote. Imagine this is the zygote. This is the zygote. Okay. So this zygote first developed into see, this zygote is developed into embryo, not directly. First zygote is developed into globular embryo. The zygote is developed into by repeated mitotic division. The zygote first developed into globular embryo. After that, this globular embryo is developed into heart-shaped embryo. This globular embryo by repeated mitotic division, then it is developed into heart-shaped embryo. Then finally, this heart-shaped embryo is developed into matured embryo. That means between zygote to embryo, this globular embryo and heart-shaped embryo first developed, then after it is developed into matured embryo. And see, listen here, one point. Whatever cells are present Whatever cells are present in globular embryo and heart-shaped embryo, that cells are called as suspensor cells. These cells are called as suspensor cells. So the cells which are present just above the globular and heart-shaped embryo, that cells are called as suspensor cells. The exact function of suspensor cells up to date we don't know. But according to the ideas, the suspensor cell push the embryo. Imagine this is the embryo and this is the endosperm. For example, just I am taking the rough diagram. Imagine this is the endosperm. What is the function of endosperm? What is the function of endosperm? This endosperm provides the nutrients to developing embryo. That's why only already I explained. Endosperm function, it provides the nourishment to developing embryo. That's why only it is called as nourishment tissue. See, listen. What is the function of suspensor cell? Exactly we don't know, but according to the scientist, according to one idea, this suspensor cell push the embryo into the endosperm. That's why that embryo, the developing embryo will get the nutrients from endosperm. But it is not exactly, but according to one idea or hypothesis, this suspensor cell push the embryo into the endosperm. Then finally that endosperm, that embryo will get nourishment from the endosperm. Clear? This process is called as embryogenesis. Development of embryo from zygote, that process is called as embryogenesis. See, listen, after the double fertilization, ovary developed into fruit, ovules are developed into seeds, we know. So come to the seed topic. Come to the seed topic. Listen. Listen. Seeds. Seed. What is our topic? Seed. Little bit bigger I am taking. Seed. So after the fertilization, ovules developed into seed. We know that one. Correct? Listen. Fertilized ovule is called as seed. Fertilized ovule means after the fertilization, ovules are developed into seed. We know that one. So that's why fertilized ovules are called as seed. The plants which produces seeds, the plants which capable to produce seeds, that plants are placed in spermatophyta. The plant, sperma means seed, phyta means plant. The plants which produces seeds, that plants are placed in spermatophyta. Example of spermatophyta, angiosperms and gymnosperm. Only. So after the process of fertilization, ovules developed into seed. That's why fertilized ovules are called as seed. The plant which produces seed, that plants are placed in the column spermatophyta. Example of spermatophyta are spermatophytes. Example angiosperms and gymnosperm. Clear? See, seeds classified into two types. Dicot seeds, monocot seeds. Seeds are classified into two types, dicot seed and monocot seed. Seeds are classified into two types, dicot seeds and monocot seed. See, dicot seeds is also called as non-endospermic seeds. Non-endospermic seeds oblique X-albuminous seeds. X-albuminous seeds also. And monocot seeds are also called as endospermic seeds. Endospermic seeds also called as albuminous seeds. Albuminous seeds also. Seeds are classified into two types, dicot seed and monocot seed. Okay. Dicot seeds in bracket, which are also called as non-endospermic seeds, are X-albuminous seeds. And monocot seeds are also called as endospermic seeds or albuminous seeds. Non means absent. In dicot seed, endosperm is absent. That's why it is called as non-endospermic seed. In monocot seed, endosperm is present. That's why only it is called as endospermic seed. Why endosperm present as or absent, we will discuss. End of the topic, not now. In dicot seed, endosperm is absent. That's why that seeds are called as non-endospermic or X-albuminous. But in monocot seed, in the structure of monocot seed, endosperm is very clear. That's why monocot seeds are called as endospermic seeds, which are also called as albumin. Albumins means endosperm only, in other words. So first starts with the structure of dicot seed. First we are going to discuss with dicot seed, very important. Dicot seed. Dicot seed, in bracket, non-endospermic seed. Non-endospermic seeds, oblique X-albuminous seeds. Structure we will discuss. Structure of dicot seed or non-endospermic seed or X-albuminous seeds. Okay. See. Imagine this is the dicot seed. Just imagine. Imagine this is one dicot seed. Imagine this is the dicot seed. So this dicot seed surrounded by the two layers. This outer layer is called as testa and inner layer is called as tegmen. Testa and tegmen both collectively called as seed coat. Testa and tegmen both collectively called as seed coat. So dicot seed surrounded by two layers. Outer layer is called as testa and inner layer is called as tegmen. Testa, tegmen both collectively called as seed coat. Means outer seed coat is called as testa, inner seed coat is called as tegmen. Outer seed coat is called as testa and inner seed coat is called as tegmen. And inside, see, inside the seed, embryo is present. This is the dicot embryo, separately we will discuss. Imagine this is the dicot seed, inside the dicot seed, dicot embryo is present. That embryo structure we will discuss separately. And see, listen, on the surface of dicot seed, one scare is present. One thick scare is present. This is the scare. It is developed from hilum. Hilum already we discussed in the ovule. It is the junction. What is hilum? It is the junction where body of ovule is attached with funiculus. That point is called as hilum. After fertilization, hilum is developed into scale. On the surface of dicot seed, a thick line is present. That thick line is called as scale. It is the representative of hilum. See, listen, with the help of this thick line on the surface of seed is called as scare. With the help of scare, seed is present inside the ovule. With the help of scare, the seed attached inside of fruit. Generally, where seeds are present? Generally, seeds present inside the fruit. Imagine this is the fruit. Inside the fruit, seed is present. How seeds are attached inside the fruit? With the help of scale. On the surface of seed, a thick scare is present. With the help of that scare, the seed is attached to inside of fruit. The seed is attached to inside of fruit. And see, listen, a small pore present in the structure of dicot seed. This pore is called as seed pore. A small pore, a minute pore, a tiny pore present in the structure of dicot seed. That pore is called as seed pore. Through that seed pore, water and other gases exchange takes place. Through that seed pore, water and gases comes in and out. Means the seed pore plays an important role to allow the entry and exit of water and other gases like O2, oxygen, CO2, whatever required. See, inside the seed, which one is present? Embryo is present. This embryo is required water. That embryo required some gases. That exchanges takes place. Through the seed pore. A small minute pore is present. It is called as seed pore. Through the seed pore, water and gases exchange takes place, or entry and exit takes place. So this is the dicot seed. Inside the dicot seed, which one is present? Dicot embryo is present. That embryo here I am explaining embryo outside separately. Very important. In the structure of embryo, one longitudinal axis is present like this. In the structure of dicot seed, inside the dicot seed, dicot embryo is present. In the structure of dicot embryo, one longitudinal axis is present. This axis is called as embryonal axis. I am writing here. Embryonal axis. In the structure of dicot embryo, one longitudinal axis is present. That longitudinal axis is called as embryonal axis. And see, listen, two cotyledons. See, imagine this is one cotyledon and this is the another cotyledon. Two cotyledons. I am giving the labeling also. These are cotyledons. So in the structure of dicot embryo, imagine this is the dicot embryo. The embryo which is present inside the dicot seed, that embryo is called as dicot embryo. In the structure of dicot embryo, two cotyledons are present. The two cotyledons are attached to the embryonal axis. Where two cotyledons are attached to embryonal axis, that point is called as node. Imagine this is the point node. So two cotyledons are present. Two cotyledons are present. These two cotyledons are attached to. Two cotyledons are present. These two cotyledons are attached to embryonal axis. Where two cotyledons are attached, that point is called as, this is the point. Where two cotyledons are attached to the embryonal axis, that point is called as node. These are the cotyledons. How many cotyledons are present? Two cotyledons. Where both attached, that point is called as node. See, these cotyledons plays an important role. What is the function, sir? What is the function of cotyledon? It plays an important role. It plays an important role to store the food material. What is the function of cotyledon? This cotyledon plays an important role to store the food materials. This cotyledon plays an important role to store the food material. To store the food material. So, because of the presence of two cotyledons, the seeds are called as dicot. In dicot embryo, two cotyledons are present. That's why these seeds are called as dicot seed. Di means two, cot means cotyledons. In the structure of dicot embryo, two cotyledons present. That's why these seeds are named as dicot seeds. Clear? What is the function of cotyledon? What is the function of cotyledon? Two cotyledons present. That two cotyledon plays an important role to store the food material. And one important point, listen carefully, one important point. See, this longitudinal axis is called as what? Embryonal axis. This longitudinal axis is called as embryonal axis, we know. See, imagine this is the upper tip of embryonal axis, and this is the lower tip of embryonal axis. This longitudinal axis is called as embryonal axis. This is the upper tip, this is the lower tip. This upper tip is called as plumule. And this lower tip is called as radicle. In the structure of embryonal axis, this is the upper tip and lower tip. This upper tip is called as plumule. This lower tip is called as a radicle. This lower tip is called as a radicle. And listen. This is the plumule. This is the node. This is the radicle. See, listen. The distance which is present between plumule and node, this one. Between plumule and node, whatever distance is present, it is called as epicotyle. The distance which is present between, plumule and node, this is the plumule region. And this is the, see. See, This is the region is node, cotyledons. Okay. So the region, the distance which is present between plumule and node, the distance is called as epicotyle. Like that, the region which is present between node and radicle, this lower tip is called as radicle. Whatever distance means from here to here, the distance which is present between radicle and node, that distance is called as hypocotyle. Epicotyle, hypocotyle. What is epicotyle? The distance present between plumule and node. What is hypocotyle? The distance which is present between node and radicle. See, this plumule in later stages it developed into shoot. This radicle is developed into root during the germination. Generally, this dicot seed germinate to form one small plant. It is called as seedling. Generally, take one seed sown inside the soil. When favorable conditions are present, within a span of 3 or 4 days, it germinate to produce a small plant. It is called as seedling. So the seedling consists of roots and shoots. From where roots and shoots are came, plumule is developed into shoot and radicle is developed into root. Means plumule gives rise to shoot, radicle gives rise the roots. So this is the dicot embryo. This is the dicot embryo. This embryo is located inside the dicot seed. It's a complicated structure. That's why we studied separately. This structure containing dicot embryo is located here inside the dicot seed. See? So this is the structure of dicot seed. This is the structure of what? This is the structure of what? Dicot seed. Okay? Here endosperm is absent. Already this is the complete structure of dicot seed, but we are not given any labeling to endosperm. Everything is there. Testa, tegmen, scare, seed pore, chiasis, water, everything you can check it. There is no label of endosperm. Why? Because of endosperm is absent. Endosperm is absent. That's why only the seeds are called as non-endospermic seeds or exalbuminous seeds. Sir, why endosperm is absent? See, simple structure, and this is the important also. See, this is the dicot seed. Imagine rough diagram. This is the dicot seed, and this is the embryo, means dicot embryo. This is the dicot seed. Inside the seed, embryo is there, and this is the endosperm. Imagine, just imagine, this is the endosperm. So initially endosperm is present, but what is the function of endosperm? Already I explained. What is the function of endosperm? What is the function of endosperm? This endosperm provides the nutrient. This endosperm provides the nutrients to developing embryo. Correct? See, listen, this is the dicot seed. Inside the dicot seed, embryo is present. And initially, which one is present? Endosperm is present. What is the function of endosperm? This endosperm provides the nutrients to developing embryo. Mean this embryo completely consume. This embryo completely consume the endosperm. Whatever endosperm is present, that endosperm is completely consumed by embryo. Finally, endosperm is not reported. That's why only it is called as initially present, but at maturity it is not. Whatever endosperm is there, this endosperm is completely consumed. Whatever endosperm is present, that endosperm is completely consumed by embryo. That's why at maturity in dicot seed, endosperm is absent. That's why only dicot seeds are called as non-endospermic or exalbuminous. Clear? Pea is the best example. Bean is the best example. Groundnut is the best example of dicot seed. Peanuts, Arachis hypoxia. Clear? So this is the structure of dicot seed. Why endosperm? This is the important. Why endosperm is absent in dicot seed? That question is very important. This embryo completely consumes the endosperm. That is the reason. Whatever embryo is present inside the seed, that embryo completely consumes the endosperm. That's why at maturity, endosperm is absent. That's why only this dicot seed is called as non-endospermic seeds or exalbuminous seeds. Clear? And in monocot seeds, we will discuss on tomorrow. Okay? Not today. Today time was also up. So we will discuss on tomorrow. Shreya Reddy, any doubts? Shreya Reddy, I repeat, Shreya Reddy, any doubt, please send me the message in group also. Okay? Thank you, all of you. Bye, Nana. Continue, we will discuss on tomorrow.